Okay, let's dive in. When you start looking deep into the human genome, you quickly find that some of the biggest health problems aren't caused by tiny typos.
Hmm,
We're talking about whole sections of DNA, massive chunks that are either missing or, duplicated.
that's exactly right. Today we're tackling copy number variants, or CNVs. These are those large-scale genetic changes, and figuring them out takes some pretty sophisticated molecular detective work. They're behind quite a few severe genetic conditions.
And we want to make this really concrete for you, so we're going to follow two specific cases.
Hmm.
First, there's Alex, a little boy dealing with muscle weakness.
And second, Coralie, a one-year-old who has this really unique, uh, almost strikingly social kind of personality, along with other features.
Yeah, and through their stories, we'll unpack three key diagnostic methods: multiplex PCR, MLPA, and array CGH.
And along the way, you'll learn about a couple of, frankly, surprising concepts. There's the reading frame rule in muscular dystrophy, which is really counterintuitive.
Yeah.
And we'll get into the mechanism behind how these large chunks often get deleted or duplicated in the first place, something called non-allelic homologous recombination, or NAHR.
Okay, let's start with Alex. He's 6. His parents notice he tires easily, he struggles with stairs, and he does that thing where he needs to use his hands to kind of climb up his own legs to stand up.
Right, the Gower sign. That's a classic indicator.
Exactly. Plus, his calves look unusually large, and the blood tests, well, they were alarming.
Let me guess, creatine kinase levels.
Through the roof,
Yeah.
like 50 times higher than normal.
Yeah, that points strongly towards muscle damage. CK is an enzyme that leaks out when muscle fibers are injured or breaking down, so those levels scream neuromuscular disorder.
And the top suspect is immediately Duchenne muscular dystrophy, DMD. It's the most common severe form, mostly affecting boys, about 1 in 3,500.
Precisely.
Yeah.
And the root cause lies in the DMD gene. It's on the X chromosome, and, well, it's a monster. It's the largest known human gene, 79 exons, spread over maybe 2.5 million base pairs. And it makes a protein called dystrophin. Think of dystrophin as like a shock absorber or structural support inside muscle cells. It connects the internal skeleton of the cell to the outside matrix.
So if it's missing or faulty.
The muscle cell membrane basically tears easily during contraction. This leads to chronic damage, inflammation, replacement with scar tissue, and of course, that massive CK leakage we see in Alex.
Okay, and importantly for DMD, the genetic errors aren't usually small ones, right? We're not talking single letter changes most of the time.
That's key. While point mutations do happen, maybe 20–35% of cases, the big players, 65 to 70%, are these large deletions or sometimes duplications involving one or even multiple exons. Whole chapters of the instruction manual are just gone or repeated.
Right, which brings us to this twist you mentioned earlier. There's DMD, which is severe.
Right, essentially no functional dystrophin.
And then there's Becker muscular dystrophy, BMD, which is generally milder.
Yes, in BMD, there's still some dystrophin produced. It might be shorter or less of it, but it's partially functional.
So, if a deletion causes the problem, why isn't it always the severe form? How can deleting part of the gene sometimes lead to a milder disease?
This is where the reading frame rule comes in, and it's fascinating. It's all about how the genetic code is read. You remember DNA is read in groups of 3 bases, right? Codons.
Right, triplets, like 3-letter words.
Exactly. So imagine you delete a chunk of exons. If the total number of bases removed is a multiple of 3, the words after the deletion still line up correctly. The reading frame is preserved.
Okay.
You produce a protein that's shorter, yes, because some amino acids are missing, but the sequence after the deletion is still correct. This is an in-frame deletion. You get a truncated but often partially functional dystrophin. That leads to the milder BMD.
I see. But if the number of deleted bases is not a multiple of 3,
Then you shift the entire reading frame downstream. All the 3-letter words after the deletion are now wrong. It's like the sentence turns into gibberish. This out-of-frame deletion usually leads to a stop signal appearing very early in the wrong place. The cell makes a very short, unstable protein fragment that gets quickly destroyed. No functional dystrophin. That gives you the severe DMD phenotype.
okay, that makes sense conceptually. But is there a real-world example that shows this?
Absolutely. And it's mind-blowing. Look at exons 43 through 46 and the DMD gene. If you delete any single one of those exons, 43, 44, 45, or 46, it causes an out-of-frame shift. Result: severe DMD.
Okay, seems logical. Delete an exon, get severe disease.
But if you delete all 4 of those exons together, 43, 44, 45, and 46, The total number of bases removed happens to be a multiple of 3. The reading frame is preserved.
Wait, removing more genetic material results in a milder outcome.
Exactly. Deleting exons 43 through 46 causes the milder Becker phenotype. It completely flips your intuition right. Sometimes losing 4 chapters is better than losing just 1.
That is wild. Okay, so for Alex, we suspect a deletion, probably out of frame. We need tests that can actually find these missing exons. Standard sequencing isn't the tool for spotting large deletions like this.
Correct. We need methods that count the copies of each exon. Now, while MLPA is really the gold standard for this gene, often labs might do a quicker screen first, especially for known hotspot regions where deletions commonly occur. That's where multiplex PCR comes in.
Multiplex PCR,
Yep.
so multiple PCR reactions at once.
Pretty much. You design primer pairs for several different exons, say 9 common deletion targets like 43, 45, 51, etc., all in the same reaction tube.
How do you tell the results apart if they're all amplified together?
You design the primers cleverly so that the amplified DNA fragment from each exon has a unique size. Then you run the results out on a gel or through a capillary system, and you can see which sizes and therefore which exons are present or absent.
Oh, okay, so it's fast. Cheaper, maybe.
Exactly. Fast, cost-effective, good for screening known hotspots. But it usually only covers a subset of the 79 exons.
Right, so for a definitive look across the whole DMD gene, you need something more comprehensive.
And that's where MLPA shines. Multiplex ligation-dependent probe amplification. This is really the first-line test for suspected DMD deletions or duplications. It's designed specifically to quantify exon copy number.
Ligation. That's different from just PCR amplification. How does that work?
It's quite elegant. For each exon you want to test, you use a pair of short DNA probes. These probes are designed to bind to the patient's DNA right next to each other, spanning the target exon sequence.
Okay, so they have to find their exact spot.
Precisely. And only if both probes bind correctly side by side can an enzyme called ligase stitch them together into a single, longer probe fragment.
I see. So if the exon is deleted in the patient's DNA,
The probes have nowhere to bind properly, or at least not next to each other, so they don't get ligated. No ligation, no signal for that exon later.
and after the ligation step,
Then comes the multiplex part. All the successfully ligated probes, from all the different exons being tested, have universal tags on their ends. So you use just one pair of universal PCR primers to amplify all the ligated products together in one reaction.
so the amount of amplified product for each exon directly reflects how much of that ligated probe was created, which depends on how many copies of the exon were there to begin with.
I got it. It's quantitative. You then separate these amplified fragments, again, usually by size, using capillary electrophoresis. You get a series of peaks. The height or area of each peak corresponds to the relative copy number of that specific exon.
So for Alex, if he has a deletion of, say, exon 45,
You'd see the peak corresponding to exon 45 either completely missing, indicating 0 copies in a male, or significantly reduced, maybe to about 50% height, if it were a female carrier who has one normal X and one X with the deletion.
that's remarkably precise for counting individual exons across such a huge gene. Okay, so MLPA can pinpoint deletions or duplications with a single gene. But what about even bigger changes?
Exactly. Let's shift gears to our second case, Coralie.
Right. Coralie is 1 year old. She has some growth delay, certain heart issues, pulmonary stenosis was mentioned,
And sometimes issues with the aorta, too.
and distinctive facial features, sometimes described as elfin-like, plus that lacy pattern in her iris.
The stellate iris.
But what really stands out is her personality. She's described as having this incredibly happy, social, empathetic nature, almost overly friendly.
That's very characteristic of Williams syndrome, that cocktail party personality, often alongside strong verbal skills, but usually some level of intellectual disability, and interestingly, hypersensitivity to sounds.
And the genetic basis for Williams syndrome isn't a single gene issue, is it? It's much bigger.
Much bigger. It's typically caused by a deletion on chromosome 7. A specific region, about 1.5 million base pairs long, 1.5 megabases, is just missing. And this region contains around 26 to 28 genes.
Whoa. 1.5 megabases. That's enormous compared to a single exon. How does something like that even happen? It feels like it should be a super rare catastrophic error.
It's significant, yes, but the mechanism is actually quite common, relatively speaking. It's due to the architecture of our genome itself. The process is called non-allelic homologous recombination, NAHR.
Okay, break that down, NAHR.
So our chromosomes are full of repetitive sequences. Some are simple repeats, but others are larger blocks of DNA, hundreds of thousands of bases long, that are nearly identical and appear in multiple places. These are called low copy repeats, or LCRs.
So segments of our DNA that look almost the same but are in different spots.
Exactly. Now think about meiosis when cells divide to make sperm and eggs. The homologous chromosomes, the pairs we inherit from each parent, need to line up perfectly to exchange genetic material, which is called crossing over.
Right, recombination. That shuffles the genetic deck.
Correct. But if there are these large, nearly identical LCRs flanking a unique region, sometimes the chromosomes misalign during that pairing process. An LCR on one chromosome might pair up with the wrong LCR on its partner chromosome because they look so similar.
Ah, like a zipper snagging on the wrong tooth because the teeth look alike.
That's a great analogy. And when the cell machinery tries to complete the crossing over based on this misalignment, the exchange is unequal. One chromosome ends up with a deletion of the entire region between the LCRs, like in Williams syndrome.
And the other chromosome.
Gets a duplication of that same region. So NAHR mediated by these LCRs is a major mechanism for generating these large structural variants, these CNVs, often in specific hotspots in the genome prone to this kind of misalignment.
Okay, so our genome architecture itself creates these vulnerabilities. Fascinating. So for Corley, we need a test that can see this huge 1.5 megabase deletion on chromosome 7. MLPA is too focused on single genes. We need a wider lens.
Absolutely. We need a genome-wide view. And the workhorse for detecting these large CNVs across the entire genome is array CGH.
Array comparative genomic hybridization, CGH. How does that work?
The array part is key. Imagine a glass slide, like a microscope slide, or maybe a silicon chip. On its surface, thousands, even millions of tiny spots are printed. Each spot contains multiple copies of a specific short DNA sequence, a probe, representing a known location in the human genome.
So it's like a map of the genome laid out on a chip.
In a way, yes. A grid representing points all along every chromosome. Now, the comparative hybridization part. You take DNA from the patient, Corley in this case, and you label it with, say, a green fluorescent dye.
Okay.
Then you take DNA from a control sample, someone known to have a normal genome, and you label that DNA with a different color dye, usually red.
Green for patient, red for control.
Got it. You chop up both DNA samples into smaller fragments, mix them together in equal amounts, and then you pour this mixture onto the microarray chip.
And the DNA fragments float around and stick to their matching probe spots on the chip.
Exactly, that's the hybridization. The patient's DNA fragments and the control's DNA fragments compete to bind to their complementary probes on the array. After allowing time for binding, you wash the chip to remove any unbound DNA.
And then you see where the green and red signals ended up.
Precisely. A scanner measures the intensity of the green and red fluorescence at every single spot on the array. If a particular spot, representing a specific location on chromosome 7, is bright red,
That means more controlled DNA bound there than patient DNA.
right, which implies the patient, Corley, has less DNA from that region than the control. She has a dilution there.
And if a spot is bright green,
That means more patient DNA bound, suggesting she has extra copies of that region compared to the control, a duplication. If the spot is yellow, a mix of red and green, it means patient and control DNA bound in roughly equal amounts, indicating a normal copy number.
okay, colors give a visual clue.
Mm.
But for diagnosis, you need numbers. You mentioned log-2 ratios before.
Yes, that's how we quantify it. The computer calculates the ratio of the patient's signal, green to the control signal, red for each probe. Then it takes the logarithm base 2 of that ratio.
Log base 2. Why that?
It makes the interpretation really intuitive, especially for copy number changes. Think about it. Normal copy number is 2 copies in the patient, 2 in the control. The ratio is 22 equals 1. The log 2 of 1 is 0. So a value of 0 on the plot means normal copy number.
Okay, 0 is normal. What about a deletion, like Corley's suspected deletion on chromosome 7?
She likely has only 1 copy instead of the normal 2 in that region. The control has 2 copies, so the ratio is 12, or 0.5. The log 2 of 0.5 is negative of 1.
Ah, so a dip down to MyGov of 1 on the graph signifies a heterozygous deletion.
Exactly. And if there was a duplication, say 3 copies in the patient versus 2 in the control, the ratio is 32 equals 1.5. The log 2 of 1.5 is roughly plus 0.6. So a jump up to around plus 0.6 indicates a duplication.
So by plotting these log-2 ratios for all the probes along chromosome 7, you'd see a clear, sustained drop to NIGAD 1 across that 1.5 megabase region in Corley, confirming the Williams syndrome deletion.
Precisely. You see the exact boundaries of the deletion based on which probes show that NECR1 signal. The resolution depends on how densely the probes are spaced on the array, but it's perfect for spotting these large CNVs.
That really paints the picture. We've gone from the very targeted approach needed for DMD checking specific exons with multiplex PCR, or more definitively, counting them with MLPA, all the way to this panoramic genome-wide scan using array CGH to find that massive deletion causing Williams syndrome.
It's all about matching the tool to the scale of the suspected problem, and these cases highlight some fundamental aspects of our genome. We saw the almost bizarre logic of the reading frame rule, where sometimes less deletion is worse than more deletion, functionally speaking.
Yeah, that exon 43-46 example is still spinning in my head.
And then we saw how the very structure of our genome, with those repetitive LCRs, creates inherent risks for large-scale rearrangements, like the one in Williams syndrome, driven by that NAHR mechanism during meiosis, microscopic alignment errors having massive consequences.
It really underscores the complexity.
Right.
From single base pairs up to mega base chunks, it all matters.
Absolutely. And it leaves you wondering, doesn't it? We know about these LCRs and NAHR hotspots now. How many other subtle variations in our genome's architecture, maybe in regions we currently think of as junk or just repetitive, are actually playing critical roles in health, development, or disease susceptibility? And how many more syndromes caused by CNVs are waiting to be discovered as our array technology gets even higher resolution?
A lot left to uncover. It makes you think about what else might be hiding in plain sight within our own DNA.